Okay, well, just a bit of background about me. I was born and educated in England. I did my degree and PhD at the University of Durham and came to New Zealand for a postdoc at Invermay Research Centre, which was MAF in those days, now of course is AgResearch. Then I joined the staff and spent time working on pasture pest management, native grassland, community ecology, looking at disturbance from fire and agricultural development on biodiversity. I've worked in invertebrate conservation on endangered species with DOC colleagues, and biological control, particularly biosafety and risk assessment, and that's what I will talk about in this talk today. So, is biological control safe? Well, after the publication of Rachel Carson's famous book, Silent Spring, published in 1962, there was huge public pressure to reduce pesticide use. She claimed that there was a persistent and continuous poisoning of the whole human environment. So research on biological control gained momentum after that, and it was widely promoted as natural and safe and risk-free. And it was claimed that there were no examples of adverse impacts, but in reality, nobody had really looked. So why wouldn't it be safe? Well, biocontrol agents are living organisms. They're self-perpetuating, they multiply, they disperse, and releasing them to the environment has to be considered irreversible. So with those attributes, there's clearly potential for adverse environmental impacts. Researchers became strongly polarised. A debate became quite acrimonious on this topic. The ecologists and the environmentalists were urging caution, and they were opposing the biocontrol practitioners, who of course were the heroes saving the world from pesticides. And there was really little research to support either position. So there was an awful lot of heat and very little light. So what are the risks? Well, the risks are mainly non-target effects. They can be direct non-target effects, where a biocontrol agent might attack a native species or an otherwise valued species. And this can, to some extent, be tested in quarantine during risk assessments. Indirect non-target effects are much more difficult, much more complex. A biocontrol agent might compete with or displace existing natural enemies, and there may be a myriad of other food web effects. So very complex and unpredictable. And there have been books written about these topics and many conferences held. So how was the debate resolved? Well, regulators all around the world were increasingly requiring risk assessment. So it was a requirement rather than a nice to know. And funding research was made available, which made a big difference and provided opportunities for some really rather good ecological research. In New Zealand, biocontrol agents are regulated under the Hazardous Substances in New Organisms Act, and this is implemented by the EPA, the Environmental Protection Authority. And the purpose of HAZNO is to safeguard ecosystems, to protect health and safety of communities and the needs of future generations. So our research focus was to reduce uncertainty in decision making for the EPA. And most of our work was carried out it has been carried out in the Better Border Biosecurity Research Collaboration, better known as, or otherwise known as B3. This is a partnership between researchers in these CRIs and end users. We work closely with end users, particularly MPI, EPA, and Department of Conservation and the primary industries. So the overall aim of our research is to minimise risk of introducing environmentally damaging organisms without potentially rejecting useful biocontrol agents. So a good way to reduce uncertainty is to improve the prediction of host range. So the case study that we've worked on is a parasitoid shown in the picture there from Morocco, which was introduced to control the lucerne weevil, a pest of lucerne. This was released in the 1980s with minimal risk assessment. This was pre-HASNO, and no non-target effects were expected. It was considered that there were no closely related weevil species in New Zealand that were related to the target. However, our preliminary studies found that several native weevils were actually attacked in the laboratory, and we found that this was happening also in the field. And the picture on the right shows a native weevil that's been dissected, and it contains a large parasitoid just about filling the body cavity. So we conducted retrospective studies. We used tests of in cages, such as shown here, as if the parasitoid had just been brought into quarantine. So we exposed a number of native weevil species to the parasitoid in these cage tests. From the results of those, we made predictions about field parasitism. But because the parasitoid was already out there in the field, had already been released, we were able to go out and carry out field surveys to verify those predictions in the field. And the EPA has been able to benefit from post-release studies such as this and help with their decision-making in the future. So another question that we asked was, could knowing the host range of the biocontrol agent in Morocco have helped us predict what it would be if it was released in New Zealand? So we identified some research collaborators in Morocco. I went over and spent some time with them discussing the project and getting them set up with some sampling, which they carried out for us over a period of about a year, sending samples back to New Zealand for us to look at parasitism in the weevils that they collected from lucerne and surrounding vegetation. And so from this, we were able to ascertain the natural host range of the parasitoid. And these results emphasize the importance of accurate knowledge of weevil phylogeny. So the taxonomic affiliations of weevils with the target. The weevil family is a large family which was not well known in higher taxonomy area, and if we knew now with methods that we can use now, molecular methods, we would have been able to predict potential non-target hosts in New Zealand, as we have subsequently found. Another approach we took to predicting host range was to look at host immunosuppression by parasitoids. This is work we carried out with colleagues at Otago University. We discovered a virus-like particle in the ovaries of this Moroccan parasitoid. You can see those in the middle of the picture there. It was known that particles like this, virus-like particles, or polydnavirus, they're sometimes called, are quite common in the ovaries of braconid parasitoids, and that they do suppress the immune system of the host so that the parasitoid can survive inside the host. And these virus-like particles found their way, in our example, into the area surrounding the eggs. And when the eggs were injected into the weevil host, some of these particles were carried through with them. But what we found was that these virus-like particles were not present in other related species that we looked at that had a narrower host range. So there was some indication perhaps that these VLPs might enable a wider host range than in other parasitoids that use other mechanisms of host immunosuppression. So there's certainly scope here for more testing of this hypothesis and in the future. So finally, some work I've done internationally. I was privileged to join the executive committee of the IOBC, the International Organization for Biological Control, and spent 4 years as president. IOBC supports and promotes environmentally safe biocontrol throughout the world. That's its purpose. And I was able to raise awareness of New Zealand biocontrol research and make a lot of contacts and collaborations that I think I've brought back to the benefit of New Zealand research. I'm still a member of the IOBC Commission on Access and Benefit Sharing, which seeks to maintain freedom to operate in the face of the Nagoya Protocol. This is a supplementary agreement to the Convention on Biological Diversity and is intended to promote sharing of benefits between countries from the use of genetic resources, actually to prevent really bioprospecting. But biocontrol agents are genetic resources, so are subjected to these agreements. And we want to ensure that biocontrol programs can continue between countries with minimum bureaucracy. So I'd just like to acknowledge colleagues in AgResearch, B3, the University of Otago and IOBC. Thank you very much.